of the lectures in the series of real fiction. Last week was like an introduction of what designers are doing in this space with microbes and bacteria and the kinds of things that are going on. And we saw two really different approaches last week. Sonia Boimel with her very, very hands-on approach, very sensual approach, working in the labs, playing with bacteria, really kind of getting in there. And then David Benkei with a very, very different approach, very analytical approach. He was sort of, although his stories were very highly fictional, they were based on sort of real speculations from sort of previous events that had happened before. So he was kind of borrowing from those events to make his speculations. So again, two very, very different approaches to how designers might start to think and work in this space. And I thought what was interesting last week was very much about how very easy it is to start polarizing the discussion, where it becomes very much about positive and negative things, good and bad. It's very easy to immediately start to set those sort of things in motion, but how we can try and move into a different kind of space, a space of dilemma, a space of sort of a contradiction, and sort of somehow feel happy in this space and explore in this space. So I think this is a way in which we can start to build thinking and sort of make more informed decisions. So tonight we have two more speakers to move the discussion forward. And these two presenters are equally very, very different approaches. But many ways they're looking at how to participate through sort of DIY bio or sort of non-expert participation, and they both use very, very different ways of doing this. So we'll do the same format again, where we have 30 minutes from the first speaker, 30 minutes from the second speaker, and then we might have a few questions this time, and then we'll have a discussion and then open up to the audience. But first I want to introduce Markus Schmidt, who is co-hosting the lecture series with us from Biofaction. So he will come and speak, just say a few words of introduction, and then he will introduce the first speaker, and Gunter. So thank you.
I'd like to tell you a little story. A couple of years ago I went to the UK for holidays, and we drove up to Birmingham, and close to Birmingham is a nice interesting village called Iron Bridge Gorge. Probably you've heard about it. This was the place of the Industrial Revolution. So what happened there is that at that time the blast furnaces were fueled with wood to extract iron from iron ore. And the problem was that the wood was in short demand, so they could only use as much wood as would grow around in the forests. So it was limited. So the output of iron with this technology that had back then was limited. And now he came a guy from the south of from England, an outsider called Abraham Darby. And his is an interesting link, a link of interdisciplinarity. So he came from the brass community. So brass community and iron community back in the late 18th century were very different. They didn't talk to each other, they didn't have the same the culture, the same traditions. So he was an outsider. And what he brought in from the brass community was the knowledge that you'd actually convert coal to coke, which is a different form of a carbon structure. And the thing was that coal contained a very high amount of sulfur. And if you would use coal to extract iron from iron ore, you would get a very low standard iron that would break down and was not very useful. So but if you convert coal to coke, what the brass community was doing all the time, and introduce that to the iron community, all of a sudden they could use the coal that was back there. So the limitations of having not enough fuel to make iron was suddenly disappeared. And that was one of the main reasons that all of a sudden you had almost unlimited amounts of iron available, which triggered a cascade of events What followed were standardizations of screws and bolts and nuts, because you could produce them in large quantities. And with this technological innovation also came a set of socio-economic changes. So I think that's also another very interesting point. That if a technology is very potent, it's not only changing the technology, but it's also changing the socio-economic fabric which we're living in. It will affect all of us. So we have seen a couple of technological innovations and changes over the last couple of centuries. There's the one in mechanics. We've seen the steam engine, the transport technologies. Electricity was a wave about 100 years ago, and we've seen the latest wave in computers the last 30, 40 years. And now some predict that the next technological wave will take place in biotechnology. So the beginning of the 21st century, biotechnology is regarded as the next big technological wave that might trigger events that will cause ripple effects in whole of society. And we have the same elements in here. We have an interdisciplinary feature, so we have people from the engineering community coming into biology and suddenly treat living organisms like a living matter that you can program. So they say, well, we take the DNA, we program it, we exchange it, we use modules, we use parts, we standardize biological systems, and so we make engineering of biology much easier. So that's the idea of a new field which is called synthetic biology. Another aim or goal of synthetic biology is to make generally biology easier to engineer. So right now, it's like in the old days where craftsmen were blowing glass. So you had to have a long tradition, a long education and training to be able to do that. So that the craftsmen and artists were very skilled in doing things, and only a short number of people would be able to do that. So if it succeeds to make biology easier to engineer, that would mean that more and more people will have the opportunity to use biology as a living substance and program the living matter. And among the people that will be then ready and able to work with biological material will be, for example, artists and designers, but also biohackers. So people that, like in the last technological revolution, were playing around with computers and software and making their own computers in the garage. So there are some people that estimate that the next, let's say, revolution or innovation wave will come from the amateurs, from the amateur biologists. So this is where the next speaker, Gunter Seyfried, comes in. He's actually also a graduate from this university, but in the last years he specialized and looked more and more into biotechnology as a medium to make his own artwork. But he also is the founder of a Viennese do-it-yourself amateur biology group called Pavillon 35. They're currently gearing up and equipping a lab outside of the university, just an amateur lab, a group of people that comes together and tries to tinker around with biology. And he has also some experience with other groups in Europe doing a similar thing. Oops. And with further ado, I'd like to introduce Gunter Seyfried and his presentation on biohacking and do it together.
So good evening. Thank you very much for having me, and Fiona just gone. Thank you very much for setting up this series of lectures, which will gear up bioactivities at this university. I'm sure for that. So in this presentation, I'm going to talk about the study on the activities of DIY Bio Europe, which I made together with Lai Pai and Markus Schmidt for Biofaction. So let me see. So synthetic biology will be mentioned in some occasions in this presentation, but Markus Schmidt can give you a more detailed account on that. Maybe for this presentation you can compare it like to Prince William and Prince Harry. So the one is a little bit smarter and put in a more responsible position, and the other is a little bit cooler but still has the potential to owe up to his bigger brother. So you can compare it maybe a little bit of that. So first I want to describe the DIY bio context a little. So here we go. So we have these three terms: do-it-yourself biology, do-it-together biology, and biohacking. So these terms describe more or less the same activity, that of amateur biology. So each of these terms tries to name a different aspect into being. So if you have the DIY bio, so you think about making, tinkering, handicraft. If you talk about do-it-yourself biology, which was coined by some groups who call their workshops this way. So the term do-it-together biology emphasizes the collaborative, interdisciplinary, educational, and sharing aspect. And last you have this term biohacking, which is kind of probably a problematic denomination, summoning this hacker fantasy banked up by 40 years of history of this, cyber warfare counterculture. So, but there's also another reference, a computer like Markus already mentioned. If you look at the history of the microcomputer industry, a look at the history of the microcomputer industry explains the euphoria that accompanies synthetic biology. Because microcomputers started too in clubs, garages, or workshops, for example, like the Homebrew Computer Club in San Francisco, when the Intel 8080 chip was brought out, and so which made desktop computers, the size of desktop computers possible. So Apple started at the Homebrew Computer Club too, like Steve Wozniak, putting together some parts to form the first Apple. So, this explains a little bit why there's so much speculation on that. But, this comparison tells more about this economic speculation than about DIY bio. So this unconventional use and research of biotechnology outside of established institutions and laboratories started as a movement in 2008 in the US. As a mixture of professional biologists, artists, designers, scholars from the humanities, engineers and IT people conducting experiments at home or in hackerspaces. So bringing in the insights from other fields. There's also this sense of dissatisfaction on how academic science manufactures reality under economic coercion. So the wish to create an alternative space for science feed into that formation of this self-empowered bio-research activities. So an alternative approach on how to handle scientific research speaks out of this wish. So first, I show you the DIY Bio Code of Ethics. I will talk about this event when these codes were created a little bit later. So you see the Code of Ethics from the European delegation and the Code of Ethics from the US delegation. So they are more or less the same. You see some minor differences, like you have in the European Code of Ethics respect, responsibility, accountability, and modesty. So you won't find this in the US Code of Ethics. This maybe lies in some cultural differences on how entrepreneurship or how technology is used. And also you see in these terms like transparency, safety, open access, education, community, peaceful purposes. So this should describe some alternative way how to conduct science. So, but we can later talk in the discussion about the implications for the society of this new technology. I think it's better than that. So, in 2009, this movement comes from the US to Europe, first materializing itself in Paris as a group later called La Palace. It's called The Bench, which is the working place of a bioscientist, and which is also the first group who had actually a full equipped and fully functioning lab at the moment. And so the latest news I heard is so they are really getting off the ground right now. So I can show some pictures of of that. So Lapayes, they could find a place in a squat. So that was the first thing they do. They hook up with hackerspaces and this squat scene in Paris to get their space. And so here's some of the entrance here below here. So here's also the next one. So this is the entrance, it's like a cellar. So here's the entrance. Yeah, you see this hackerspace kind of look, tinkering on electronics. But if you go here right through the corner, you get to the actual lab they set up. So you have some washing the dishes is mandatory there too. So you have some cyclers and heating devices. So ingredients, cooling. Yeah, I think this was kind of a sterile workbench. And here's the actual bench where they could really conduct experiments. So here's the... So actually this is a leftover from the company. There was a private company who was taking care of the water in Paris. And this company went bankrupt. And from there, they could obtain a lot of equipment. So this is one strategy from these groups to get to cheap equipment, like seeing what's left over in the bankruptcy, scavenging equipment in the internet, and stuff like that. So this group was followed by Biologie Garagen in Copenhagen in 2010. I can show you some pictures of them too. So in the studies, we tried to interview 10 groups. Oh, I'm sorry. Okay, we tried to interview some 10 groups, but 5 agreed of them, and we were able to visit 3 of them. So I showed the pictures of that. Oh, I forgot this one. So the group in Paris was well organized, so they could set up this first European-wide meeting where all the groups from Europe could meet up, and some of them see each other for the first time. So let me start here. So this is Thomas Landrain, the founder of Lapayes in Paris. So this is Ketel Garvey from Ireland. Actually, we were not able to interview him, but he's actually the first guy who got the full certification for his bedroom. So he's really allowed to make GMO experiments inside his house. So he's kind of a lone wolf guy, but he offered a lot of possibilities to the other groups, and writing down protocols that can be used. So this is Peter van Boehmen from the Dutch DIY Bio. He and 3 friends of him invented the Amplino device. It's a qPCR, a quantitative PCR machine. The PCR machine is to copy bits of DNA. So with the tinkered device, he is able to detect the quantity of, they started with malaria, so it is part of malaria device. So this is really a big field that DIY Bio gets into, because there are a lot of innovations that are left behind by big companies or by big pharmacy or big governmental institutes. So sometimes it's too expensive, to pay a postdoc, to spend 2 years like soldering something which may never work. So, but they made it, and they really have a great success on that. So, and this device could be used for different stuff too. It can detect all kinds of DNA. So, if you want to test your meat or any other kind of food, it could be used for that too. So here's more of that convention. So there were around 50 people from all over Europe attending to the first meeting. So I'm sorry for the switch pictures, was just not my God, what's going on? Okay. So 4 big projects were set up during this meeting. One was the bug idea. So in your backyard, there are a lot of bacteria and organisms that have special kinds of features like antibiotic features and stuff like that. And this is one project to build up a database with all these organisms and see their functionality and open it to other groups who want to use them. Okay. So it was always there was also a big discussion on legal aspects. Also, problems of European integration came into play because what might be or what could be allowed for one group may be forbidden for the other group in the other country. So a lot of problems around European integrations are in this European-wide DIY bio movement too. Yeah, let's see what else we got. So this is another project, the Biohacker Starter Kit. To make a small starter kit so that everybody can get involved in DIY bio and starting to doing their own experiments. And one was the DNA communication systems, just to decode and encode messages with DNA or proteins and send them around to the other groups, to make own communication system. And one was this one, I forgot. So this is a Bio 3D printer. So yeah, I'm wondering if if this will going to happen. So I'm very excited about this. So let me see. So after Paris in 2010, there was the group Biologie Garage in Copenhagen. I can show you pictures of them too. Let me see. Okay, here we go. The entrance is a little bit similar. So Biologie Garage in Copenhagen was founded together with L'Habitat, the hackerspace they are in now. So the founders, one of the key founders, he was also the founder of L'Habitat. So to make a DIY bio space inside this L'Habitat was in the concept right from the start. So you see some plant experiments. Here they have in the next one. So, and you see they also have very sophisticated equipment, but it's not cleaned up, as you see, and it's not built up properly. So here you can see one of the problematics if hackerspace and the biospace is combined, because normally, since the hackerspaces have been there first, so it's always a competition around resources and space. So and mostly it's in the favor of the hackerspaces. So you see the same thing in the Dutch DIY bio. I can show you the next one. Oh, I forgot too. So at the time I've been in Copenhagen, the Biologie Garage together with StudioLab and the Medical Museum set up a big series of workshops and exhibitions. So I just happened to see the build-up of the DIY Biolab in the Medical Museum. So this is Rüdiger Trojok, a guest biohacker from Germany who makes his master, I guess, in Copenhagen, doing some electrophoresis here. So this is how actually a biohacker lab looks like. So this is the gene gun from Rüdiger Trojok. So he built a little gun with air pressure that shoots small pieces of DNA inside of cells. So this can be a alternative and cheap way to put DNA in other organisms. So it's not very effective, you know, you have to try a lot, but sometimes it works. Okay. Yeah, that's the thing about pretzels. I found out this afternoon that it cannot handle, you know, other formats than 4:3 or 6:9. So, so we have it here. So, and the next one is the Dutch DIY Bio. So right now they're located in the Waag building. So this is the Waag at the Neue Markt. It used to be a marketplace, and the scales have been in this building. So the scales which ensure, how you say, I forgot the word, Gerechtigkeit? Justice, you know, that to ensure justice on the marketplace, that you have the right weight of the right quality of the products and the right price. And so that's a symbolic coincidence. So what ensured justice in the 16th century, you know, in the same place, there are now these fab labs and hacker spaces who try, you know, to reinstall justice in science too. This building also bears the anatomical theater, you know, from the famous painting from Rembrandt. You know, the anatomy, our lesson from Dr. Tulip. So there have also been executions on the Neue Markt. So when convicts were executed, they were carried up in the Teatro Anatomico and dissected there right away. So just setting up the workshops. So this is an open PCR machine. This is a low-cost PCR machine, which was one of the most wanted perks that these groups want to have. So they have really for $500, they could have their own PCR machine. Now setting up the electrophoresis. So there were also filmmakers present. So they have a lot of attention in Amsterdam for that. So, like I told you before, the problematic about, you know, this competition around space. So this cupboard is, you know, designated for the biohacker group, while the Fab Lab has much more space. So, but you see, this is what I before called do-it-together biology. So all the participants, you know, sit together in groups and work out materials and devices and equipments. So this is the start of electrophoresis, an open-built electrophoresis. So this is Teatro Anatomico I was telling you before. So, and what you found out is that there are important building blocks to set up a DIY Bio group. So, the founding members of 3 groups participated in the iGEM competition, which helped. So the iGEM competition is a synthetic biology competition where professors or group leaders send their undergraduates, where they, for the first time, can really experience and learn how science is done and how science works. So another building block is institutional assistance, like from Genopool in France, where it's one of the biggest bioparks, just like we have in Vienna, like Marx, this biocluster. Another one is the Waag Society. I already showed you in Amsterdam. So in the Waag building, there's the Waag Society located. It's a society to propel art and science in combination. So, like I mentioned before, the connection to established hackerspaces like Temp Lab in Paris, Fab Lab in Amsterdam, L'Habitat in Copenhagen proved to be helpful in the beginnings, providing space, small fundings for workshops, and some kind of license to proceed further in that field. Though, like I told you before, it became visible that this collaboration also created a competitive situation around resources in the favor of these hackerspaces. But not only the impulse came from the US DIY Bio, also two major events worked as a catalyst for a European-wide movement. So the first one was the DIY Bio Continental Congress in London on the 8th of May 2011, organized by figures of the US DIY Bio scene, where the groups of Europe met for the first time. But it was still not a European-wide meeting, but some of the groups who were there at the time met for the first time, for example. And the second significant event was the DIY Bio FBI outreach on the 12th of June in 2012 in San Francisco, where European activists were invited, which left an odd impression on them, actually. So that's kind of ironic. So the two major events to set up or to form from these loosely networking European groups to a European movement were set up by United States, by the FBI and by the DIY Bio groups there. So especially the event in San Francisco let's see. Put really the energy forward for the European groups to create their own discourse, taking control over the shape over their activities, because it's like if you can't forbid something, power structures intend to use a discourse to control the course of events. So that was some kind of a try, to put this discourse into action and to have control over the European groups. And somehow they had this feeling about this, and they really tried, to shape their own course. So. But it appeared, so these were some major factors for the formation of this group. But it appeared that the most significant factor was personal interest, passion, commitment, a professional background, and leadership qualities. All the possibilities derive from this individual qualities, interacting within the communities, networks, and the public sphere. This individuality also contributes to the unsettled character of DIY Bio, resisting conventional definition and formalization. And you find this all over the groups. Like I showed you before, Thomas Landren, he's a PhD student and he's really a professional in biology. And I think he would be like way up high in the conventional, conventional institutional science, but he decided for himself, to set up an alternative to that. You can also see it in Copenhagen, where Mark Juhl Christoffersen, Lasse Nordfeldt, and Malte Borch founded this group, and Mark Juhl Christoffersen works now in the US and is an expert on this alternative tinkering kind of biology. And you see this Peter van Bohemen, who developed with his friends the Amplino device. So this was a strategy for them to have this clear mission around this Amplino device, from where they got a lot of attention, and which helped to bring out this group, because they have more than 100 inactive members, but who are very interested in what's going to happen in Amsterdam too. So DIY Bio may not be that amateurish like the common notion suggests. You see all these professionals set up the groups, although it is open for amateurs to learn and maybe develop own projects. So interesting and promising discussion and collaboration between experts and non-experts from other fields are starting here. So the founders of these groups are disclosing the world of biotechnology for others. And this could be you, if you hook up with a group like that. Thank you very much.
So actually, Koby started life as a graphic and communications designer, having quite a reputation designing books, and then joined the Design Interactions program at the Royal College of Art, where he got an MA. He also lectures in visual communications, but has been involved in many different design research projects, from Microsoft Research. He's been working at StudioLab-Design, so doing a lot of work with moving into this area of symbio, and I think it's very interesting. Koby's taken a very, very different approach to how designers might participate. So I'm not going to say much because I think we'll pass it on to you, and we'll find out in the discussion. So thank you.
Thanks for having me. So basically, I think in this context of like real and fiction, I think I'm going to talk about just one project, and I think I'm going to try to show how reality, basically, to me. Is it okay? So basically, you can hear me? Sorry? Okay. Sorry. Okay. So basically how facts and reality, I kind of converted them into a fictional work, and then how basically life itself took these fictional things and kind of took it back into reality again. So that's kind of a really strange, distorted relationship between the two. So it all kind of started last summer, basically when this is how I started my graduation show, and where it kind of was published that an artist genetically engineered a mice with the DNA of Elvis Presley. And they shouted it that it just required 3 online services which anyone can use. So basically a project that I started as a really innocent, poetic, maybe philosophical project, suddenly became, when it was published, became to this really horrific horror movie scenario, which I didn't kind of anticipated. And we called it the Elvis Gate. So it all started with a kind of a project I did in my first year, which I called Transfiguration. And I thought about this idea of genes as poetic things. So basically, each gene represents a story in our life, or each gene can actually code for a story, in our biography, in a way, if you can want to look at it that way. I'll pass this thing. So I decided to take my grandfather and to try to kind of revive him, just by taking some of the stories I knew about him. So I kind of looked at, his childhood, things I knew about his family. He immigrated, and I tried to kind of translate it into different traits, kind of behavioral traits. So like intolerant, he's a pessimist, he's anxious, and I looked at genes that actually code through those things. And I discovered that there is such thing as transgenic mice exists. And I thought, that would be great. I can take some of his genes or some of those stories, and I can just put them in a mouse, and this mouse will suddenly, be my grandfather. And I took it one step further, and I decided I'm gonna design my grandfather's life, or the potential of this creature, of this transgenic mouse, become my grandfather by actually putting him into different scenarios or different stories, or create the environment that will take this genetic and make it a sort of a real life story, or a real biographical. So this is basically how it looks. It was this kind of a modular system. Every combination was a sort of another story. something related to his childhood, something related to his death, something related to a traumatic event in his life, and it was all kind of built with different sort of small components, basically kind of components of, of life, if you want to look at it that way. The end of his life. And when the project was finished, I kind of I look at the way I really want to develop this project further, and I look at what I kind of accomplished in this project. And I think from the critical point of view, I really like this idea of us abusing or using technology for for our psychological needs, which is another need which I think is really relevant and which we have to kind of approach. And from the philosophical view, I think it was really, kind of a starting point to kind of touch of how we actually define life or how we can represent life. Sorry. Yeah. And from the scientific view, the whole notion of kind of nature versus nurture and the way kind of science uses models as a way to look at reality, or when you kind of really vague of what is reality and what is a model of, or how come models become our reality, and so on and so forth. And what I wanted to avoid basically was this idea of it was too personal. It was about my grandfather, and no one really had no one cares about my grandfather except me. And it was a bit too poetic because I just used a bit of science and that's it, and started kind of developing a story about it. And it was too cruel, kind of using this mouse, kind of presenting this idea of a living mouse actually living in this models. So my new motivation was actually to provide the crowd a nicer, easier point of kind of easy way to get into the project and to relate to it, to understand some of the parts that I was talking about before. So half a year later, I don't know if I was drunk or whatever. Basically, I came up with what I thought at the time was a really great idea. So if it's too personal, I'm gonna try a public figure. I mean, everybody gonna know, like a celebrity. And if it's too poetic, I'm going to take a DNA, because DNA, it's like an empirical truth. And if I got a public figure, everybody can identify with it. If they hate it, if they love him, if they hate her, or if they love her, and everybody would believe it basically if it has like a sort of a scientific foundation. And I came up with the idea of getting like a DNA of public figures. And how do you get DNA of public figures? You just go to eBay, which is really great because I found like this really flourish community of people selling like celebrities, pre-owned items. Like I started up with a t-shirt, a sweaty t-shirt by Pamela Anderson, and there were letters with stamps licked by I don't remember, like those criminals, and really rich a rich platform to kind of look for those things. And when I came up and kind of after researching a lot, I came up with those three figures that always came up again, which are basically three mega cultural celebrity icons which died before their time, and probably in terms of our culture, we kind of made them as sort of gods, and we want to revive them, and we want to have them here. And we're going to do everything that we can to actually to maintain them, living. So I bought a hair of Elvis Presley, and I bought a hair of Princess Diana and JFK in eBay, which they sell it around 20 pounds, or I don't remember the exact sum, but it was something around this range. And you get those certificate that this is the hair of Elvis Presley, took from his barber this year, and those are the signatures. And so basically you get those hair, and you get something that is signed from behind, which is a certificate that says that this is the hair of those celebrities. at the time, yeah, I was kind of so I really want to prove that, you know, I can actually take the hair, I can sequence it, and I was looking for different services that can actually do it. At the time, there was, I think it was the beginning of 23andMe. It was really expensive. It wasn't £99, and there were other services that that kind of offered a forensic test. So send us a hair, send us a stamp, send us a T-shirt. We'll get the DNA from it. We'll sequence it. We'll give it back to you. But it was really expensive. So I approached those two scientists, which I can't show their faces now, but we'll talk about it later. And we made this really kind of a mini proof of concept, where we took one of the scientists' hair, and we tested for actin-3 gene, which is the athlete's gene, which says if you have a potential of being a really, really good athlete. The scientist didn't have it. And you can see it in the red circle there, the mutation. So basically what it means, just to have a nice illustration, what it means in terms of mice, that you can basically have a mouse that can run for hours. And he beats all the other mice. So that's kind of the parallel into, you know, those a gene that you're going to take from here, where you're going to find the parallel in the mouse model. So then I came into kind of how do I approach it from a design point of view of what am I actually going to do with it. So if I have the hair, I have the genes. I kind of divided it into two. One, I'm going to have to kind of read everything I know about, everything I want to know, everything I can know about Elvis Presley. And I kind of divided it into those really serious biographies and Elvis for Dummies, which is amazing. You have to have this book. Which kind of, and I started kind of dividing it to different, you know, the most important aspects of his life that I thought would actually mean something in a mouse life. And the other side was the more scientific part, where I basically bought a book that every scientist that uses mice as a behavioral tool has to kind of look at what those models means. And I set up with this, like, I think like 50, 60 models, I don't remember exactly. And every day I kind of took this biographical info, and I tried to see maybe that would fit with this experiment, and that would, you know, that would create a mouse that would be more anxious, and that would be, and kind of made a sort of distilled Elvis's life basically into 6 different phases. Which was, one of them was his childhood, his talent, one was about his self-image or public image, commodity, which was the Graceland period, and the death. And I kind of, this is sort of a retro thing, but I kind of imagined it as something like, you know, like that. So taking this fact that he was a mama's child, so he's basically growing in a small chamber on the first floor. He has to, he was bullied as a child, so there's like a behavioral test that actually introduced a bigger mouse to a smaller mouse, which kind of makes him really afraid. And there's the idea of making a lot of effort to get his food. So basically everything kind of happens in those models or in those cages. Supposed to kind of create the right environment for him to grow into being Elvis with his genes. So the next part was kind of how do I construct it, and I started kind of dividing everything to really small pieces. I really like this idea of kind of making life into different parts and kind of assembling it together and disassembling it together and disassemble it. And so I started kind of make this really a modular system, kind of built from metal, and I can just tweak it every direction I want to tweak it. If I want to create this environment or that environment, and it looked something basically in the beginning something like that, like different modules, chambers, and it was just the collection of those things. and started kind of looking at the environment that he kind of came from. And this is like some pictures from Graceland. I kind of thought about colors and maybe materiality and how it kind of, what it has to kind of show. And this is how it looked in the end. So basically the whole sort of statue is based on sort of a linear timeline, which starts with his childhood till his death, where he goes from the bottom chamber into the top chamber, basically having the life of Elvis Presley as a mouse. To communicate it, basically I knew that I don't want to use the living mouse. So what I did is actually use the sort of a specimen just to illustrate the idea of having more than one mouse kind of going through this system, and saying if two mice with the same genetics, what does it mean in terms of having the same circumstances, the same life circumstances? Would they actually act or behave the same? Would they actually be the same, or would we kind of perceive them as the same entities? Those are some. So, as soon as the project ended, I think I was really happy. As like every student, yeah, it's in this blog, and they're talking about it in here. It was really nice. I was really happy for a couple of days. And then came this nice email from Wired, which he wanted to make a sort of an article about it, and he started asking me questions. And I started getting like those warning signs, which I decided to ignore. But it's really nice to see the process, I think, kind of from this point of view now. So basically he was asking me, was it difficult getting the medical staff to create Elvis mice models? And I answered, no. At this point we did a sample test with one of the scientists. So kind of I'm answering just, instead of saying, no, I didn't use these things, I'm just saying, no, I mean, this test doesn't exist, we're doing something else. And then he asks me, what were the names of the two genetic services you used? And I'm answering, there's tons of those. This one, and this one, and this one, and this one. And that was another warning sign that I kind of, I didn't use. And after a while, he kind of published, they published an article which said that I actually did a mouse that is Elvis Presley, and it's amazing, or it's really awful, or it's really good. And it started like, it started to scare me. I mean, after a few hours, and I got those two different opinions. And one of them said that, yeah, this is what's happening with the press. This is what they're going to do. Just stick to your thing, and everything's going to be okay. And the other one was, Koby, it's really scary. Think about what you're doing. Contact them. Do something. And while I'm kind of thinking about it, it was all during a weekend. This is what happened. Basically, it got like everywhere. Suddenly, people started kind of blogging about it and doing like those crazy Elvis mice and Elvis fans and science blogs, and it became like this one of the top trendy stories of the week. And I got really, really scared. I mean, this is something I didn't do, and everybody are like talking about something I didn't do. But at the same time, like really nice things happened. So I kind of discovered, like, it was just a few days after, Madonna suddenly kind of pick up her DNA. She doesn't leave anything behind. So Madonna do it, I did Elvis. I mean, there's something here that I'm really, I'm on it. Yeah, it's really great. And suddenly I'm getting this invitation to be in a board, something called Lifeboat Foundation, which like a foundation that's going to save the world, and with Ray Kurzweil in the same thing. And I'm like, really, I'm starting to enjoy it. And I think one of the most amazing emails that I got was from a company in the US, which deals with animals, and they're actually asking me to buy the clones of Elvis Presley to manufacture them in the US. And I was shocked that it's actually, I mean, for me it was really great because, you know, I said, wow, amazing. I mean, this thing kind of gets there. It's making what I'm, you know, kind of part of the things I really intended it to do. But with all this kind of craziness and nice things, I suddenly got an email from the scientist I was working with that says that there's a company that was mentioned in the article basically threatened to sue the people I was working with from it was Imperial College. Wait a second, sorry. Yeah. So basically what happened is that there was an article in the Mail Online that says those things, and this company kind of read about it, and they said, no, how can it be that we're doing those really unethical things? We don't have a record of a mouse that is Elvis that someone kind of ordered from us. And how can you help those, you know, those really crazy projects that are kind of going in there? So it kind of started a really, I think, a really, for me, it's kind of made me understand what's the, you know, the relationship between just being a designer and, you know, printing this thing and sending it out. You know, people read it, don't read it, it's really okay. Then actually making something that can, you know, has a lot of kind of responsibility in it in terms of what it can do and how you have to kind of maintain those really delicate relationships. So in the beginning, I was really, you know, enthusiastic. I'm doing what's right. I'm fighting for freedom. They're doing those things. I can say whatever I want about them. But then, you know, it got really, really complex in terms of what it means and what it means to those scientists and how science works and how we work and how we can kind of maintain those relationships together. So basically for the next month, I spent, instead of kind of, you know, talking about the work and just taking it one step further, I was just kind of trying to take everything that was kind of written in the wrong way out of the web, basically, which was really hard. Some of it, you know, just like the Daily Mail kind of erased it. Some of them changed it. And it was really a hard time. I was sitting in my room, like basically like the mouse in the cage. Just you know, the police is gonna come and shoot me in my house. And I did this. I had a plan of, you know, the personal apology, the public apology. You know, I kind of published this thing in my website. I sent those press releases to everyone that kind of published something about the project. And with a lot of help from the RCA PR department, we kind of managed to take it down, or kind of. So I think in the end it was really nice. I got a really nice cover, which was like, for me, was kind of fixing everything. And I had this interview with someone that. I think every 2 sentences I told him, you know that this thing is not true. You know that this thing is not true. So basically, I think one of the first sentences in the article was, it's worth emphasizing that the Elvis mice do not exist. And finally, it was like, yeah, finally someone kind of talks about this thing in the, you know, in the right context. And a nice thing that I also discovered, I think after the fuss, is like, I think it was a few months afterwards, one of the services that was mentioned there, the forensic services, they deleted this, basically to offer a forensic test in their website. So basically the idea of just sending them, you know, someone's belonging is, you just can't do it anymore. So you can just do your own thing. So the context of taking someone's DNA and just send it doesn't exist in there. And I think I kind of took it, you know, I kind of found myself with this project talking about it in, you know, in science, relating to science, related to design right now. And I think this is what I'm still kind of really enjoy the project, because I think it kind of managed this nice relationship between this facts and fictions, kind of maintain it in a really kind of in different environments that that I really like, because you have the opportunity of kind of explaining it in this context and sometimes explaining it in that context, and it's kind of produce a really nice environment to kind of you know, produce it or produce more. Yeah, I think, yeah, I still have time. Or I talked myself too. Yeah, I mean, right now I'm working with the StudioLab project with the RCA, with the department of the Design Interaction department. The project called Blueprints for the Unknown. It's kind of a research about different implications of synthetic biology, and I'm kind of trying to take this idea of you know, the things that are there, the things that are there for us to kind of use. What are they actually used? What we can actually use them for, or what happens if we kind of mesh them all together in this really weird combination? And what I basically did is kind of mapping a lot of different services or a lot of different technologies and you know. Movements that we can, you know, kind of, we're witnesses now. So there's like, you know, the idea of open genetics, that people are just sharing their genetics in a sort of a social networks. And this really nice website called OpenSNP, which kind of allow people to research the relationship between phenotypes and genotypes, and by sharing everything they have about themselves. So what I am trying to do with those things is actually taking a service, if you know Cleverbot, which is artificial intelligence, that is out there for almost, I think, 10 years, talking to people, learning from people, and I'm taking this weird personality that for me kind of represent the reasonability of you know, of people, all the people in the world talking to this one thing that kind of learns from them, and I'm trying to actually give it a sort of a biological representation by using everything that is open source and crowdsourced. So if you look at this artificial intelligence, using the OpenSNP, kind of looking at its phenotypes, you can use something like the Genome Compiler, which is a software that basically synthesize together different genes. You can send those different genes to a DNA synthesizer that was built from Lego in the iGEM competition in 2009 to produce basically DNA, or to produce proteins. But it's all speculative, of course. It's not real then, no. And using those DNAs, you can actually print an organ, for example, by just building yourself a reprop that uses bio ink. So the whole kind of mesh-up of those things, kind of taking an artificial persona and make it and give it a sort of a biological representation, that's what I'm trying to kind of work on it now. It's all sketches, it's all just in the middle of kind of making, but yeah, that's the general idea. Yeah, thanks.
I would like to ask initially about regulations, because I think it's interesting. Hello, right, that's it. So I'd really like to know from both of the speakers about how regulation, what do you think, how regulation and limitations, what kinds of regulations in both the areas do you see as shaping the next lot of experiments that you do? You know, what regulations might come in and how that might shape the kinds of experiments you do. You know, what role does that have?
I think from my point of view, I mean, it's. I think I discovered as soon as the project, as I kind of finished with the project, that it's not legal to sequence someone else's DNA in the US and in the UK. That's for sure. But there are countries around the world, which is probably most of the countries, that don't even kind of started getting into those legislation, and basically you can do whatever you want if you kind of outsource it to wherever you want and just get the clone, get the new creation or whatever.
Yes, the same way, because in Europe you already have very strict regulations, so basically not everything is allowed here, especially in the DIY bio sector. So if you have a accredited lab, it looks different, and like you said, all the major labs do it the same way. If there's a regulation they're not allowed to, they send it to a lab to India and get their, for example, animal experiments there. So, so I'm not sure if there are some more regulations coming to that. So there are already strict regulations on that. So it should be more, maybe, like I told you, it's in Europe. There's a lot of fragments of regulations. So to sit here.
Just last week I met Thomas Landré, who is the founder of the Paris Do-It-Yourself Lab. And for his latest project, he actually had in mind the regulations, and he did not want to do any genetic engineering. So his idea, his plan was to do a pen that contains a bacterial medium with bacteria, and that would produce blue ink, so he could write by feeding the pen with sugar and water, just like that. So the easiest way would to genetically engineer any bacteria. But he was actually going to the database and was looking for naturally occurring bacteria and found one that occurs in the soils of South America, and you could buy them from a database from one of these retailers legally, imported it. It's not toxic. The ink that they produce a pigment which is blue and is non-toxic, and the bacteria is also a low risk. So actually using natural occurring bacteria that have the same features as he would want to do with the genetically. So actually by taking into account regulation, he found in the natural biodiversity something that suited very perfectly what he was doing.
Basically, that was about the project from the Europe-wide movement. I showed you, this bug idea, to get organisms from the backyard, that do specific stuff, which are already there and already have this trait. So it's completely legally to get the bacteria out of the garden and let it do something.
It's also, I think, the question of. It's not just the question of the legal thing, it's also the question of who's monitoring it. I mean, when will be the time that you're going to find like a police officer that will come and say, sorry, you did something illegal? Because right now, I just remember, in our studio, we had a student with no name, which ordered like cockroaches from Madagascar. And we had those, it's basically illegal, but it got through eBay in an envelope, and who knows where they are now, somewhere in the UK, and kind of messing about with all the the relationship between things.
Do you think that's something that affects what you think you'll be able to design? I mean, are you shaping your thinking based on regulation? Does it shape the things that, so in the biolabs in various places, are they aware of the things they're doing in relation to regulations or not? Is there sort of an awareness of that or not?
Also, it doesn't influence my thinking, because the lack of my bioregulations knowledge allows almost anything in my thinking. But when it comes to collaboration partners, so most of them are in an established laboratory, and they have their liabilities, so they just cannot do what they want. But like I said, there are some ways to get around that. Maybe sometimes they will do it if it's a really good friend. He will do it for you, but then you cannot go to the public with it.
I think it's a good idea because it's a very interdisciplinary endeavor. You have people from the arts and also scientists and all these amateur biology groups, and at least one biologist in there, and someone who knows about regulations a little bit and safety. So it's always good to have a friend in the group who's looking over your shoulder and and pointing out if you're doing something that could hurt your own health or that of your friends and family, and point it out. So in order that you don't have to, of course everyone is dealing with this stuff has to get some basic knowledge about it. Because if you have an engineer and artist dealing with genetic engineering, of course you have to get some idea about the biosafety and biosecurity as well. Because it's a self-replicating device, not like an Arduino or a computer that you can throw away. These things actually can get out and replicate. So of course you have to take that into account.
I'd also like to ask about what questions do you think both of you are asking that are not being asked within a kind of conventional science domain? So, you know, what are you looking at that you think is different from the conventional approaches?
Yeah, like the example I showed you, the Amplino device. This is a perfect example for that. So I was also talking about this economic coercion. So, you know, these established labs, they are really under big pressures. You know, the team leaders are under big pressure, so they have to propose for grants, and they have the liabilities, you know, and they have to take care that the next year they get the money too. So there's a lot of pressure on them, and so, you know, they won't look on the right, on the left side. They're just happy that they just made it to you know, secure their fundings. So like the Amplino device, there are a lot of innovation that's on the way. You know, like I told you, it's for a well-established lab, it's maybe too expensive to have a postdoc who costs a lot of money, you know, to give him 2 years' time or 3 years' time to soldering some kind of this device, which will be very cheap and replicable at the time, you know, like to sell, maybe to help third world countries or stuff like that. But these are the kinds of innovations that are really, you know, left aside from big companies, and you know, because there are vast amounts of money in that bio research. So there's a tough battle about this money, and so I don't think there is any way around this. So and this is really the point where DIY Bio can get into the picture.
Basically, I think one of the things I'm trying to do that doesn't happen in science kind of context is the idea that there are more things than just kind of saying we're going to save humanity with this thing, and we're just doing a research to save humanity or research to cure this thing, which I think this is how they kind of show science, but in a way, I think 50% of science is sheer curiosity, and you can't get anywhere without this 50% of curiosity, or just, you know, doing things because we're really interested and we think something gonna come up, but we don't know what it is. So sometimes it's just because we're really interested, sometimes because we think we want it, and I think I'm really like this this kind of hidden part that they kind of live, you know, beneath the surface. That is, it's there, but nobody basically admit it.
I also have a question. There are two other former graduates from the RCA who also were misrepresented, misinterpreted by the medium. One is Tuur van Baarn. He was at an art show, Next Nature, and on stage he allegedly modified bacteria in the yogurt, so they would produce Prozac, an antidepressant. So within a couple of days, you could see all different newspapers all over the world showing pictures of depressed scientists and then eating a yogurt, and they would cheer up all of a sudden. And I don't know. So in a way, it seems that this misunderstanding doesn't really hurt the designer artist. In a way, it actually propels the career of the artist, because suddenly your name is known, your project is known, and although you declare, well, that's not what I wanted to do, it's incorrect, but actually you actually in your career it actually brings you forward. So is there any good reason to really try from the beginning not to be misunderstood in this tough world where it's hard to be recognized as an artist? I ask this as a provocative question.
I don't think. I think the whole idea is just kind of doing it, because you know, I'm coming from an environment where you just, you know, you have those ideas and you just do them, and I think thinking of those implications is something that you can, I think this is why those things basically happens in the beginning of your career, because you don't know about what are the implications could be. And you can't listen to it. I think I didn't listen to anyone in a way, because first it was really hard for me as a student to listen. I mean, it's something, I don't want to listen. I want to do what I want to do. And it was, I think, part of my kind of personal, I don't know, getting against anything. So if someone will tell me, don't put your finger in the electricity hole, I will put my finger in the electricity hole just to see what happens, because I really need to do it. So I think this is the idea of, you believe in it, you really get really interested, you don't think about anything else except this project, which looks like, the most amazing thing in this period of time, and that's it, and everything else kind of disappears. And so I don't think I think I'm really sure that it wasn't intentional. The only intentional thing I can think of myself kind of while doing this project was the idea of choosing a celebrity as an entrance point to the project, which I think I was struggling a lot with kind of, communicating this idea. How would people, understand why a mouse? How can they connect to this, to this mouse, to this really strange thing? And I said, yeah, I'm going to use, Elvis Presley, or it would be really easy in 2 seconds. Yeah, I see Elvis Presley. What's happening with Elvis Presley? Why is a mouse? Why? So I think this is kind of the thoughts that
Yeah, I guess a lot of provocation works just over the media right now. It's not like 40 years ago, when we have some, we have the Wiener Aktionismus, when you have those kind of art events, it was a whole provocation for the society. But right now, it's just only works over the media. So if you want to be provocative, it just works over the media. And so, and this is another. When I told you the 3 terms, do-it-yourself biology, do-it-together biology, and biohacking, when you name different aspects into being, but it also has some consequences. Biohacking is used by DIY biologists, to allure the media, or to have some bait for them, and the media use it, to lure customers for the newspapers. So this kind of vicious circle, like from me getting media attention and providing media attention. So, but you have this whole different kind of fantasy or imagination then after that term. So if you use do-it-together biology, it has this, this collaborative education, but biohacking, it could be good, could be evil. You have all those fantasies like you had in thousands of movies, like it's a whole culture about that, hacking into high security networks of the finding out evil government conspiracies or getting killed for doing that, and all this kind of, but it has nothing to do with the actual synthetic biology biohacking itself, because it's more about finding out, and we don't know about the consequences when doing so.
Does that mean, though, that you can only do certain experiments outside of a conventional setting? Because obviously, if there are certain things within as partnering up with conventional scientists who, they're going to be very worried about working with people who are sort of pushing the boundaries. And does that mean that kind of activity stays completely outside of conventional science, or do you think there's any opportunity for sort of more provocative work to work with conventional science? Because from what I can see is it's almost like they push each other away, and the conventional scientists don't want anything to do with anything that could be taken up by the Daily Mail and then sort of makes their science look bad, and then they could be seen as funding gets cut and all the rest of it. So I'm just wondering about what that relationship would be between conventional science and then the experimental things that try to sort of push out and ask provocative questions. Because another thing I've also noticed is that you can't do anything that has any negative connotations within a professional science. So it kind of limits in some ways the dialogue that's going on. It has to be within conventional science. So for me, it's about what is that relationship, and can that relationship between conventional science and experimental science, can they come together and do things somewhere, or does that just water it down?
Yes, I guess, if conventional, non-conventional scientists or meet for a nice dinner, I think they're right at the same place. But when it comes to the professional place, the conventional science, like I told you before, there's a lot of money in it, and it's very rationalized place. So where there's no space for art, for fantasy, or even for crazy ideas. So sometimes it takes a lot of guts, and it needs to need to be a very self-confident scientist to allow an artist within his lab, to just perform what the artist thinks is going. Because
I think basically, like you said before about kind of the Industrial Revolution. Basically, it's the same. I think it's kind of the same situation when you got artists kind of starting communicating with people from the industry, or artists learning about working with the not technician but not the people who designs machines, the people who work the machines, the workers basically, and you have those people who knows how to tell stories and have creative mind, and you have the people who knows how to operate things and knows what's good, what's wrong, and suddenly I think there's like a new way. It's just the beginning of this new situation where you get those people with all the rules, all the methods, and everything kind of really well constructed, and the mechanism, and you get these crazy people that try to we can make we can take this thing further, or we can think about new things, or we can we can suggest and you get the same it's just like modernism in a way, just a new wave of this thing, and a new wave of relationship between, I think, the industry, which I see as more of a scientists, and designers, which so this is.
I think it's like a circle, So conventional scientists, can open up a world for artists, and show them a whole new array of possibilities. But like it's on the other side. But it also, what we found out with the DIY Bioscene, it depends on the personalities. You need really a strong personality to be a receiver of that vision, in either art or in the field of science.
I think there is maybe a conventional science, but then there's also an unconventional science, and probably one of the best scientists, they like to think in different ways. So they are probably the ones that are more open or more interested in an interaction with artists. For example, at the lab in Harvard Medical School with George Church, they have an artist in residence, Joe Davis, and he's one artist and 70 scientists, and that's what George Church said, probably the best ratio, because otherwise they would not be able to work, because he just brings such a chaos into the group. But 1 in 70 is okay, so he's pushed around. But it's a good influence of thinking outside the box and looking at things from different ways, but just in the right mixture, the right solution and blend. So they actually help the scientists and in this interaction find something new. And also there's the first question. There's
Who needs a mic?
in English, please. So I talked to a scientist, and he wanted to do an experiment which in the field of biochemistry was seen as something that would never work, so he would never get funding. But in the context of an art science experiment, he could do it because he would get back some liberties he had lost in the scientific game. So he was actually using, in a way, this artistic angle to make an experiment he would otherwise not be able to do. So it's a kind of claiming back some freedom that is normally assigned to the artists.
We've seen an example or two of speculation about what could happen in such a world, which is what you showed. But I would love to hear another example of something tangible that actually came out of one of these labs, because we've seen a lot of images of dirty labs and heard the word DIY bio like 50 times or more. But apart from that machine that you hinted at once in one of the photos, any other examples of crazy stuff that came out of these labs, even though it's such a new movement, something exciting?
But it's not 100% made in these dirty labs. It's the bulletproof skin from Jalila Saidi from the Eindhoven group, Bioart laboratories in Eindhoven, and she mixed the spider silk gene with the skin gene, and they really grew the skin in the in the petri dish, and then went to the shooting range and see if it's really bulletproof. And I think for small caliber it worked. So this was one thing, but as you see, if it's that complex, you can be for sure that parts of it have bing done, with specialized equipment in a really high class, how you say, laboratory. So what at the moment really comes out are these educational features, doing these workshops. There were plenty of workshops in Amsterdam with a Dutch group, like they had these urine testing workshops, which were quite fun. They also had, a performance by CJ, by Charlotte Jarvis, when she donored, the cells from her. But when she worked on that further, to really make stem cells out of their old cells, it needed a highly specialized laboratory. So I can only tell it for Europe. So this Ampline device I showed you this. So this is the most advanced thing that came out of that. So you can still use the gene gun I showed you as well. So you told me once in the Soviet Union, when they tried to mimicry these genetic techniques, and they couldn't have this sophisticated genetic engineering. So they tried it with a gene gun like that, just firing thousands of times, and suddenly they got 2 cells picking up that DNA, what normally was manufactured in a secret lab somewhere in the United States. So it's really hard to get this kind of examples who can match themselves, with an output of a lab, because it's right now starting. It started in the United States in 2008 and came to Europe in 2009. But like I told you, like in Paris, it looks like a dirty lab, but they're really getting off the ground, getting some major funding from somewhere else, and you can see some of the results maybe in the next 2 or 3 years, I guess.
I think that the work that you see in these DIY biolabs normally gets overestimated, never underestimated so far. So the level of technical complexity that can be done is still very low. But it's not only the technical sophistication. There's also another aspect. Is this asking different questions and trying to get on it from a different angle. Like, for example, a couple of years ago in the States, a group of DIY bio people collected fish samples from sushi restaurants to see if the fish that was actually sold as the fish was really the one or another one. And of course they used professional equipment and laboratories to find out what kind of fish that was. They needed some markers, but it turned out, I think in 30% of the cases, that the fish was another one, so a cheaper one. Yeah? So they were actually on a large scale, actually a food scandal, if you want, but nobody cared. So in a similar way, when the horse meat issue hit Europe, I mean, the first thing that comes to mind is, well, what else is out there? And so DIY people like that could go and sample and look for stuff, and I'm sure you will find something. I mean, there was even the case in Iceland where they had a meat pie, and they were looking if the meat pie contained some horse meat. Turned out it didn't contain any meat at all. For some reason. So I think if some people go out and ask questions that are not done by the bureaucracy or by someone with the power and by the elite, then you will find all kinds of interesting answers.
So these labs, they right now they only show a potential that could have been, but like you said, it shouldn't be the technical complexity that is the innovation. Like in the Amplino device I showed, it was technical devices that have been around for more than 20 years. And nobody had the idea to make a cheap malaria diagnosis for the third world until a DIY bio guy came. So that's some major innovation from the point of view, for a lab in somewhere in the jungle. It's a great innovation, for a guy here, and it doesn't matter.
Actually, 2 weeks ago, I saw a Twitter from Bill Gates who said, innovation doesn't always need inventions. Just by taking things that are already there and applying it in a different way, you can come up with a whole lot of different new insights.
The question is whether you were disappointed, or it seems like that your whole project, like the cage tower, was actually reduced to the mouse in the jar.
In the beginning, yeah, that's how they kind of showed it. I mean, this is the mouse, this is it. You don't have to imagine anything else. And for me, it was just, look at this thing, imagine the mouse. So, yeah, I think I was really... I don't think I was disappointed. I was really afraid at that time. It was really a frightening experience for me. Right then came, I think, the disappointment, because it suddenly just moved from a place that I wanted it to be, sort of, as a design object, as an art piece, or something kind of sits in this environment, and suddenly it became this really kind of a yellowish press feature that, everybody just, this is crazy, or oh no, this is horrible. So it kind of reduced everything, for me. But in a way, it kind of created a nice space for this thing to, exist in, which I think it should, in a way, in terms of just showing us, you can actually do this thing. Would you actually do it or not? So I think makes sense. Yeah, okay.
Why do you think it's happening now? What do you think? What do you think is making people interested in science in this way? Because in history there have been like other technological advances or big areas of public interest, like the space race, but no one really tried to build their own rockets and send them up into space. Like, why do you think with the biotech thing this is happening?
Yeah. So like I told you before, it's how to name something into being, because, guys who are playing with bacteria alone at home has always been, or even guys building rockets, small. There's a DIY bio rocket project in Copenhagen. So, or if you think of the first inventors, like Benjamin Franklin or guys like that, that they really made great inventions at home, like. So, but now that there is a movement, you call it DIY bio or you call it biohacking, then if it's even more to it, I have this famous example from Hubert Dreyfus, and he said, like, that there was for a long time, people were using drugs and sitting in a hot tub, and suddenly, in the 70s, it was called the laid-back lifestyle. So suddenly everything fitted together, and then it was more to it. So I guess the recent developments in synthetic biology and making biology more accessible, because before you never had access to these labs, only if you knew some guys or anything. Now the lab comes to the citizens, so it's more accessible. So, and if a certain amount of people getting involved in this, it's suddenly it gets the break-even point, or how do you say it, where it really kickstarts and grow. It was the same with the computer industry, because there were only computers in big companies, like insurances, like military, stuff like that. Nobody had access ever to the computer, but there were hundreds of guys who had the lust, to get hands on this kind of computers. And when the first Altair, came out, it was like a kit to build yourself in a computer magazine or in a DIY magazine. It suddenly, a whole industry started, and now you have web designers, graphic designers, and all this kind of stuff, starting 30 years ago with some guys, putting some chips together and suddenly had a computer at home. So it's always how to name something into being, then it's more to it.
There are actually these amateur rocket science people that meet each other, for example, in the desert of the United States, and they fire these high-powered rockets into the sky. So there's a whole amateur science movement in very different fields, even astronomy, with they buy themselves big telescopes, and they're looking for the next comet that can be named after them. So I think that there are a lot of amateur science activities going on. I tried to get a couple of books from amateur biology. The oldest one I found was from the States from the 1960s. So actually this has been around for some time. But I think, yeah, as you said, together with the synthetic biology hype in a way, this gets an additional fuel for burning a little bit longer. And they get also the media attention, the way they're looking for some new stuff that is in a way outraging, it's a little bit illegal, it could be like the next big thing. And biotechnology is preparing itself to be the next big thing for the economy, for societies also. And this is also a bit daring to do something which is actually a little bit forbidden. And so I think that a lot of, from the media point of view, it's a new thing, but actually stuff like this is going on for quite some time.
But I actually also think this whole thing about 3D printers, now you can get 3D printer, but now designers are designing their own 3D printers, and they don't want to buy the one, they want to design their own and make their own artifacts from it. And it's almost this idea that the kit technology is evolving much more. Now there's a kit, a bio kit for schools, and the idea that you could even mess with this stuff and start to do it is something that we didn't have before. You felt that you had no access to work in this way. There was, it was left to only one group of people, a specialist group. So opening up that space, I think, it is a sort of a number of things and factors coming together. Any further questions?
It's just like there was this group of kids that sent like a Lego spaceman to space. You saw this YouTube? That was frightening. I mean, it's just like a Lego like person kind of, but it got into space. So it doesn't matter. I think those things doesn't matter what you do with them. It's suddenly like this really like this magical thing that you don't know anything about it. It's really I don't know how it happens, and you're always afraid of those things. I mean, because you just don't know. I think.
From the Felix Baumgartner Lego man, no? With a
just saw
I also want to talk about just briefly the idea of biohacking and the relationship between the media. I wonder if somehow the term citizen scientist feels unthreatening. And I'm wondering, if the case of people using science to sort of regulate or participate collectively to find things out is not seen as sort of biohacking, as opposed to I'm wondering if by changing the way that people participate by using the term citizen science would create a different kind of movement, and what would that mean to sort of democratize the technology more? I mean, any further thoughts?
I mean, if you look at the earliest hacker movement that came out of the MIT, there were a bunch of nerds that they were probably quite smart, perceived themselves as super smart, and they were proving themselves all the time that they could make the same thing with fewer codes, and it was a kind of a competition showing off who's the smartest guy in the room, and this smartness radiated, so everybody wanted to be as smart and as has the same kind of social esteem as these guys, no? So I think the hacker community goes along this line, that there are people that are super smart, they have the insight and they can check things out and they understand everything they do, which is super cool. And on the other hand, as you said, there's this citizen science, which is not as presumptuous. It's just a way to be more inclusive, more participatory, and take people into account from, even if they're not that extremely bright as the super hackers, but there's a way to let people participate in new endeavor. I think that you're right. It's a total different social framing of getting people into this field, no? Making it an even more elite stuff that it already is, or letting people get on board from all walks of life. And so I think it doesn't matter. And the other thing is, this do it yourself is also quite like you, yourself, me, I, the ego trip thing versus do it together. That's something you can share. And also the whole thing that biology is something that you could do open source and open access way of doing also comes from the IT sector. And I think this is also a more inclusive way which is very interesting, and probably in the long term this inclusiveness will be probably more successful. Although you need, on the other hand, also this light bulbs that attract these people into the field.
Yes, and also many years of consumerism. So right now, you know, we're in a time, you know, where you want to get back, you know, because you just everything is presented to you, everything is laid out for you. So very systematically. So you want to get back, you know, make it yourself. You know, there are a lot of hobby cooks right now who may be quite as good as normal chefs, you know, who really try to be the best cooks among their friends, you know, and showing off with their cooking skills and inviting everything else. So this is in many sectors, you know, just to empower yourself again. It's also on the political scale, you know, you see a lot of these initiatives, you know, more democracy here, more participation that, and, you know, we don't want this power station, and, you know, we want to keep those old nice buildings and stuff like that, and they start to, you know, to gather around and take things, you know, in their own hands. And I think now it's a good time for that. So because it was a long time, you know, where nobody cared. It was just caring about, oh, where does he get the next stuff? And now is, how can I build the next stuff?
Can I ask you a question? Basically, it was always kind of interesting to me, thinking of, you know, hacking. You have those computer hackers who basically try to kind of, you know, go against the establishment and prove that they can, you know, they can hack a bank. And they actually do it one day, and then kind of, you know, it's a show-off. So what would be, you know, the equivalent in like biohacking? I mean, would you have to, you know, what would you, I mean, what's the kind of the last, you know, boundary in terms of proving that you can actually do it? You know, you can actually change something, or kind of showing the establishment or the scientific structure, you know, that we are better.
I mean, there would a lot of stuff to come up with. You know, one big thing would be if you could make GMOs without using genetic engineering. You know, just like your setup. It's just setting a whole, you know, parameters of a specialist environment, and for example, make the bacteria, you know, change their DNA according to the environment. And so it couldn't be again referred as a GMO and could be done by everyone. So if you really can make this chemical evolution get going in a DIY bio context, that, you know, that would be quite an affront to the established science. Then the other thing is, you know, if you, because in the United States there is I didn't talk about that too, because in Europe it's not such a big thing. In America is that medical aspect for DIY bio is really a big thing there, you know, to test your own DNA for certain illnesses, find cures for different kind of stuff. For example, if you find a cure for multiple sclerosis or for ALS or any, you know, the disease Stephen Hawking has or anything like that, I mean, if it would come out from such a sector, it would, you know, would quite make some shockwaves into the established science if somebody come, hey, look at this.
I think that you would have to prove that you understand organisms to a really, really deep level. I wouldn't, honestly, don't expect that from the biohackers, but from some really smart scientists that like to go a different way. For example, by changing the core metabolism of a cell, or by reducing the number of genes or metabolic pathways necessary to run a cell, by making it simpler and simpler, or change it from one kind of core metabolism to a different one, and create something that has never existed on Earth. I think if you can achieve that, it's similar to what you said, to change something on a very deep level, that would be kind of a feature that would be impressive. But I would not, as I said, not expect that in one of the underground labs. These have more of an educative approach, or yeah.
This is to Koby, and what for you is, as an artist, the piece now? Is it still the installation that you made, where it was looking at the possibility of science, or is it also all the aftermath that came with it, which is looking at something a bit different?
I really want, in a way, I think for me it would be great to kind of now put the science a little bit kind of aside related to this work, because the science did kind of destroyed me in a way. So now it's just kind of enjoying the I don't know if it's the aesthetic or the more kind of poetic part of it. So now it's kind of had a bad experience with the scientific thing, and now I just want to relax with the kind of more poetic thing. But that's kind of a more personal, kind of a more, I think, just more personal. But I think I really want to kind of push it more into the kind of design aspect of it, I think, and to show the relation of kind of how science kind of interlaid into this project, not just the science of the project. Yeah.
Well, if the amateur, or in your case, the artist, is the better scientist, or let's say the more innovative or exciting or interesting specialist, what does that mean for education then? What is the environment to engage this freedom or empowerment? No, I mean, and that questions, specialist education in a way, doesn't it? I mean, that applies to designers, to scientists, biologists.
Specialist in what way?
Well, I mean, in your profession. In this sense, if an artist comes up with an idea that even convinces scientists in a way, or the power of media managed to do this, but inspires for further thinking. So what does this mean actually for the whole system of sort of education in a way?
So yeah, I think in many ways that it's really important that educational processes do sort of open up to more cross-disciplinary practices. And maybe it's a case of...
I mean, maybe it's also something we talk about, artistic research. How sort of, does it apply in the real world of science and in the real world of design?
But I think that's exactly what we're trying to do, is look at how within an educational process can these new sort of components fit together, within bringing people in, the outsider, into a different world to sort of stimulate and bring new material in. So I do think that, I'm not answering this very well at all, but I think there is a role within an educational process, not just as a, to use research within educational frameworks to create new knowledge through learning. I mean, that's just what education should be anyway. But in some ways, I think, even with our students, they're doing a project at the moment which looks at microbial world and what does that mean, and the questions that they're asking are quite different and thought-provoking, and the way that they're now thinking about how objects change and how we relate to objects in different ways. Maybe on the end of the lecture series we could probably show some of the work that's coming out and some of the questions that are being asked during that process of learning about bacteria and what it means to designers. Because in some ways, I'm not, we work with so much materiality that to then work with something you can't see, and how much can we actually manipulate it, but how much can we imagine it, and what's that boundary between the two things? The proof of concept is always a kind of problem, whereas if you go into a workshop and you can make it, it's there and it's physical and it sits there. But if you've got bacteria and you can only imagine it, a bit like what Koby's doing, but how can you then sort of know that it's real and sits in the world, and so it shifts. It completely shifts of how you interpret and evaluate that work. You know, it sits in a very different place. So I think a lot of it's about trying to find out what the mechanisms are in that space. That's something I think we're doing now in the same way as the biohackers are trying to work out what are things they, you know, how to work out that space. I think in education, how does all these disciplines come together to work out new questions through design is what is something happening now, I hope.
I think there are many narratives. So one is a lot of my friends, most of the friends, they work in a field which they are not educated or specialized. You know, this means it could be an education in digital art, but he works now as a graphic designer. So the boundaries, they try to melt down between the disciplines. Then on the other hand, the other narrative is that the complexity, how you say, or to confront problems, for example, in society or in technology. So there are huge overlaps over the disciplines. So if we're now at the stage where the problems cannot be approached just by one. So you need a biologist, and then you need a physicist who explains the biologist how the molecules spin and stuff like that to build new devices, for example, in this electronic microscope and stuff like that. So it takes, like in the common American movie, you have the submarine and then you have the sports guy, then you have the nerd and you have the other guys with all the different skills, and they, in the end, they could, manage, the task they were front from. And so you have these huge overlaps, and I'm not sure if education reacted to that already. It is good to right now to create specialists like this, but like an approach like Fiona has in her class, to bring in other contexts is the way to go in the future. Just, not that you yourself as a designer, a specialist in biology, but then you are some kind of empowered to ask the right question to the biologist, and find a common language. I think for this way it's good. You still need that specialist, to really go deep into the subject. But then you need some other guy who can be as an interface between the specialists, I guess.
I mean, for the broader societal questions, there are not just the scientists that work in the lab to think about it. There is also like the whole technology assessment community, looking into the societal ramifications of their work. But the designers and artists, I think, are an additional way of introduce an additional way of looking at things that is can be quite valuable. But I think it's the more valuable if they actually do also a little bit homework and try to get into what's really going on in biology to understand and to be more precise and in addressing issues and asking questions. So in a way, it's also a co-education in many ways, and what it means for education, I think that as long as the disciplines operate increasingly on their own, I think we have a problem. I think there needs to be more fertile interaction between different disciplines, and that may help all of the disciplines and all of the involved people all along. But as I said, I think everybody has to do their homework. You can also see bad examples of interdisciplinary work, and that's something to be avoided.
Okay, well, I think we should finish now. So let's give a round of applause to our two speakers and thank them very much indeed for their presentations. And our next lecture will be on Thursday the 2nd of May, and we'll have science fiction writer and critic Bruce Sterling speaking with Markus, and I think that should be quite an interesting one because we really are looking at the idea of the role of science fiction and public participation, citizen, the idea of citizens and the public imagination, where you know, for the public imagination, how those two things come together. You know, how can we use science fiction? How far does science fiction help us imagine new kinds of futures? And where those boundaries occur? So please do come to that in 2 weeks' time. So, okay, I think we should finish. So thank you.